structural proteins. So whatever you are watching here, all are nothing but structural proteins. So the structural proteins are very much important to characterize the virus, right? So for example, here what are the structural proteins? Capsid is a structural protein, and again we have embedded proteins known as virus attachment proteins. Those are nothing but a structural protein, and again here we could see matrix proteins. These all are structural proteins. If you want to identify or characterize a virus, first you should know what are structural proteins and what are non-structural proteins. I will come later what are non-structural proteins. Please go ahead. The next, here see, in insects, a particular type of virus forms a proteinaceous matrix known as polyhedra. Polyhedral bodies we could say. This is single virus. All these virus to produce a matrix protein, this is known as polyhedral body, as you could see here. So this polyhedral body will give you a protective cover to the virus. What happens basically, if the virus is exposed to sunlight has UV rays, right? These UV rays are very much dangerous to virus capsid layer. But if the virus is in this particular polyhedral body, they can withstand temperature, they can withstand pH and humidity also. Any polyhedral forming virus can withstand up to 2 years in the natural environment. So this type of virus we can generally find in sericulture. If any virus is forming such type of structure, we are calling it as accalcion body or polyhedral body, as you could see here. Now symmetry. Symmetry means what happened? In the second slide, in the structure of the virus we have seen, virus is nothing but a nucleic acid. So nucleic acid surrounded by a capsid layer. How this capsid layer is arranged? Based on the arrangement of capsid, we have 3 different kinds of viruses. The first one is helical. This is rod shape. This is nucleic acid surrounded by protein, capsid protein. In the form of helical fashion, we are calling it as helical. But sometimes you can see such type of structures, geometrical structure, we are calling as icosahedral. Okay. And this third one is complex. So there are only 3 different shapes of viruses we can see in the nature. One is helical or maybe icosahedral. The third one is complex set of virus. So almost all the viruses which affect silkworms comes under this category, icosahedral as well as helical in nature. Now, if you want to start any research, it is very much important to collect the literature. What is there in the literature already? So we have to look for the literature, compile the data so that you will understand what is the research so far happened, what are the gaps and what we have to do. So as for I already told you that most of these wild silkworms are susceptible to virus diseases. So obviously if I want to do research, I wanted to know what type of work so far happened. So we have collected literature. Based on the literature, we have classified the pathogenic, insect pathogenic viruses. As you could see here, occult virus, non-occult virus. So what is meant by occult? As I already shown, if the virus is forming a matrix protein and inside it will reside, that is known as occult virus. Non-occult means just a nucleocapsid. And again, I told you that like humans, plants, animals, viruses also has its own genetic material. What is our genetic material nature? What is our genetic material? DNA, right? Virus is also having DNA. There are some RNA viruses, again in turn single stranded RNA, double stranded RNA. So based on the genetic content as well as the structure, we have 4 different kinds of viruses over here. So one is double stranded DNA. This is a baculoviridae, we call it as. The second one, double stranded RNA, we call it as reoviridae. And then we have single stranded DNA and single stranded RNA viruses. As you could see here, this it's a yellow color, cypovirus and inflavirus. This particular information actually from our lab we have generated this. This is the overall picture of different viruses we can find in the sericulture. Yeah, this particular table will tell you different kinds of viruses, the disease and the corresponding crop loss. Now, I will tell you, as I already tell, these anterior species are known as wild silkworms. They are susceptible to different kinds of viruses, diseases. As you could see here, their symptoms. This is domesticated one. As you could see, there are 2 different viruses. One is a bidensovirus and the nuclear polyhedrovirus. Whereas here in Assamensis, we have cypovirus. In Myleta we have iflavirus, and priorly we have again NPV virus. Now I will tell you, these are the characteristic symptom of virosis in Muga silkworm. So earlier we used to get symptoms like this. We were not knowing which virus is causing this particular disease, and this is the one of the costliest silk in the world. Okay, 80% crop loss. That means that once this disease comes in the rearing field, you will lose 80% of your silkworms. Only 20% silk will survive. So we know this is a disease, but we don't know the causative agent. So now the research starts from here. We want to identify which is the virus which is responsible for this particular disease. So what happened? As you could see here, first this is the control one. You can see progut. This is the gut, mid-gut part of the insect, and you could see the virosis affected one. You can see kind of ulcers, and also entire mid-gut is actually calcified. There looks like white color. Okay, this is a hallmark of the virosis symptom. So as you could see, these all are dead worms. These all are dead worms because of the virosis. They are falling down. Okay, these are the healthy worms. Would you be able to see the figures? Can anyone see the figures? Clear? Okay. Now why we want to identify which virus is causing this type of mortality? Because as I already tell, one single silkworm cost is around 10 rupees now, right? So you could see how much crop loss is a farmer is facing over here. So the first step is, we know this is a virus. So first and foremost important step is we have to go for microscopic analysis. The first one is light microscopy. So in the light microscopy we can see polyhedral bodies. These are polyhedral bodies, this round color, right? This is a scanning electron microscope followed by transmission electron microscopic studies. So viruses are very ultracellular organisms we could not see by our naked eye, right? So we have to through TEM through only we can able to see. This is transmission electron microscope. This is the midgut portion of the insect where you could see different kinds of polyhedral bodies. So this is at 5 microns resolution. If you can go further, if you can result 0.5 microns, this is the polyhedral body. In between we could see virions, individual virions. Here it is very clear. You can see these are the dots. Each dot corresponds to a particular virus, single virus. These all are virus. Don't make noise, please. Okay. Yeah, you please go ahead. Then, if you could compare this structural comparison, we have two different viruses. One is NPV. So earlier we have reported this virus. You can see these are the rod type structures. But here, which is we are finding in the Muga silkworms, we don't have this type of rod shaped structures. Instead, it has polyhedral bodies, including have a small dots. This dots indicates a spherical icosahedral virus. So in the classification of the virus, we have seen already. Go ahead. See, they are forming polyhedra. That means that they are occulted virus. So no need to do any studies regarding non-occulted virus. We have to focus only this particular portion. Now in the occulted virus, the TEM, transmission electron microscope, clearly indicated that this is not baculoviridae, because baculovirus forms rods, but it is showing small dots. So which virus form a dot-like structure in the transmission electron microscope is nothing but reovirus. But this is just a structural morphology. We want to prove molecular-biologically, yes, this is a reoviridae or cypoviridae. Go ahead. Then what happened? We have extracted, first we have purified the virus, extracted genetic material. As I already told, like humans, virus is also having genetic material. The genetic material may be a DNA or maybe a RNA. So once you extract genetic material, immediately we will come to know whether it is a DNA virus or RNA virus. As you could see here, we were unable to extract DNA from the purified viral particles because this is not at all a DNA virus. We used to get only RNA. The RNA looks like this. It's a segmented RNA, not a single molecule. There are 11 different segments will be there. So this is a segmented RNA occulted virus. Like this now slowly we used to gather the information regarding this particular pathogen. As you could see here, this is the RNA. It's not a single molecule, segmented RNA. Later slides I will show you. Then what happens, since we know the information, this is RNA virus, then we have designed different kinds of primers to amplify structural as well as non-structural genes. What are structural genes? Structural genes actually encodes structural proteins, right? So whatever bands we could see, these all are structural proteins. One is non-structural protein, that is RDRP, RNA dependent RNA polymerase. Like humans, we have DNA. Our DNA is replicated by DNA polymerase. Same thing, virus is having what? RNA. RNA has to be replicated by an enzyme that is known as RNA dependent RNA polymerase. So then what happened? We know these bands are there, but we don't know what is the sequence of these bands. We have to clone these bands, then send for sequencing. Based on the sequencing information, we will come to know which type of structural protein is this, which type of RDRP. So all this homework has done, and we have to submit all this sequence into database. Then it is found that this is nothing but a SIPO virus. It is occluded virus, double stranded segmented DNA is the genetic information. It is having RDRP, that is RNA dependent RNA polymerase. Please go ahead. Now see, there are 22 different kinds of SIPO virus. So far we came to know this viral pathogen is a SIPO virus. It is fine, but which type of SIPO virus is this? There are 22 different kinds of strains are there. Each virus has their genetic material is different. All are double stranded RNA, all are segments. You could see, for example, SIPO virus 1, it forms segments like this. SIPO virus 2, it forms segments like this. If you can see SIPO virus 20, it forms segments like this. So all are double stranded RNA, segmented genome, but the segment size, their electrophoresis pattern is different. By simply extracting genetic material and see in the database comparing your electropoietic pattern, we will come to know that which virus it belongs. For example, let us see SIPO virus 1 over here. All the SIPO virus are forming same kind of banding pattern. Yes or no? See similar kind of banding pattern. SIPO virus 14, again same. That means that there are 22 different kinds of SIPO virus. All viruses have double stranded RNA, all are segmented, but only particular group forms particular pattern. All the SIPO virus here we have same pattern, whereas another one different patterns. For example, ours, please go ahead. So this is Muga silkworm, which is infected by this double strand RNA virus. As you could see, this is the banding pattern. This exactly same banding pattern we can find in the literature of the SIPO virus which is affecting Antheria mylitta, that is tassar silkworm. And exactly again it is showing Antheria proili, oak tassar silkworm. What does it mean this? The virus which is infecting in Muga silkworm, the virus infecting other wild silkworms such as mylitta and tassar is same. They are all having same genetic pattern, right? So go ahead. And again, now you came to know this, all the viruses is same. We want to find based on the RNA banding pattern, we came to know this virus is SIPO virus 4. There is one more method to identify that. I told you that this is a segmented virus. For example, let us see, these are different segments. All segments, for example, this is a DNA RNA molecule at 5' end, at 3' end, there is a conserved region. Every virus has its own specific unique signatures at 5' and 3' end, right? Every nucleic acid has a definite polarity. 5' is there, 3' is there. So we have to look at what is there, what is the sequence at 5' end, what is the sequence at 3' end. For example, you pay attention on this. This is CPV1. CPV1 at 5' end we have TTA GCC. So if you find TTA GCC in any at 5' end, simply we can call it as this is SIPO virus 1. Why? This particular sequence is unique to this particular virus. Similarly, you see another SIPO virus 22, what is there at 5' end? AT-AGC, this is different. So we want to now investigate what is this regions lying at non-coding regions. What is the conserved regions at 5' end and 3' end? So go ahead. Yes, for that what you have to do, for example, this is RDRP, RNA dependent RNA polymerase. This is a non-structural protein, very much essential for replication of the virus, because virus is having RNA molecule now. So now we want to investigate what is the sequence at 5' end and what is the sequence at 3' end. So we can occulted virus. The virus not a rod shape. The virus is a spherical icosahedral. The genetic material is double stranded RNA. However, it is not a single molecule. It is a segmented molecule, and each segment has this particular signature sequence. Based on that, now it is finding that this is a cypovirus 4. So basically we have ICTV, International Committee for Taxonomy of Virus. This is the apex body in the virology in the international level. They actually define the virus based upon this. So as for ICTV taxonomy also, the virus which is affecting Muga silkworm and responsible for huge crop loss is nothing but cypovirus 4. So now first we have identified the pathogen up to this level. Hello, then let us see the relationship between cypovirus 4 with other cypoviruses. So that what you do, we have to develop a phylogenetic tree based upon this RDRP molecule. As you could see here, cypovirus 1, all cypovirus 1 form same clade. They infect Bombyx mori, Lemintera disper, Dendrolamus punctatus and Dendrolamus sibericus. These all are lepidopteran insects. These all are actually agriculture pests. Okay, there are 22 different kinds of cypovirus. The cypovirus 1 infect this. Cypovirus 4 has so many insects. It is going to affect. So that's why all 4 will form in one clade phylogenetically, because during the evolution all have come from the same ancestors. That's why in the same clade we will have all the things. So look at here, cypovirus 4. It is forming this triangle, as you could see, triangle. This is cypovirus 4, which is affecting Muga silkworm. Is have 100% similarity with Antheria mylitta. That's why in our genetic RNA pattern, they are having similar kind of segments, similar kind of electroparogram, right? So based on this, we'll come to know this. Now it's very clear. Cypovirus 4, which is infecting Antheria assamensis and mylitta, is similarly related. Further, the cypovirus 4 has close relationship with cypovirus 22. That means cypovirus 4 and 22 has something is, something is there in the evolution. Both have come together, right? So such type of information we can run. Then this is a kind of heat map, because while you are drawing such type of a phylogenetic tree, we are having RDRP sequence, and we align RDRP sequence, we find how they are similar or dissimilar in nature. Then we can see such type of structures. We are calling it as famously heat maps, where the blue color indicates that all are same, yellow color indicates that they are distant from each other. For example, let us see here, cypovirus 5 this is cypovirus 7, and this is something around cypovirus 50. So you can see more yellow color in this particular isolate. That means that among all these 22 isolates, these 3 particular viruses are very much unique. They are not at all showing any relatedness with another cypoviruses. That means we need to investigate their evolutionary pattern as well as their etiological studies. Yeah. Then, so now we have identified the pathogen as to be cypovirus 4. So similarly, during this protocol, we have developed a protocol. For example, if any scientist to work on cypovirus, or you want to know which type of cypovirus he is handling with, so we have developed a protocol like this. Whatever you have seen, first extract the polyhedral bodies, see under the microscope any polyhedral bodies are there or not. If polyhedral bodies are there, then extract the genetic material, and you have to find whether it is a DNA and RNA. If it is RNA, then again we have to develop electroprogram, and we have to compare this electroprogram with the existing database. Parallelly, we have to identify the conserved region, signature sequences at 3' and 5' end. Okay, then you develop a phylogenetic tree. All together, finally, if you can generate information, all this thing, finally we can identify the virus at strain level. Please go ahead. Now, the first step is identification of the pathogen. The second step is to develop diagnostic methodology. Sorry, we have developed a multiplex PCR. What happened? These wild silkworms are susceptible to not only viruses. Along with the viruses, there are other diseases. For example, let us see here. There is one virus, Ifla virus. There is pebrin. This is one pathogen. This is also one pathogen. The third one is viruses. Generally, if you want to identify virus, we do PCR. at a particular time you can identify only one